Okay, welcome to week 6 of Psych 101, General Psychology. This week we're talking about sensation and perception. Sensation and perception. Okay, today we're just going to talk about sensation. We won't get to the perception part, but there's still some fun things in there. This is all the stuff we're going to talk about. Okay, the sensation part today is what we're going to get to. Perception will be on Wednesday. So let's just get started for the sake of time. There are a bunch of slides and I like to get it done in the amount of time we have. So let's define sensation. What is sensation? Okay, sensation is merely the experience of sensory information from your senses. Okay, so here's a technical definition for you guys. Sensation is the stimulation of sensory receptors and the transmission of sensory information to the central nervous system. So sensation is merely basically when your senses detect something. So your vision, your hearing, your smell, your taste, and touch, those are your 5 senses. They all sense things from the environment, or they all detect things, okay? All of them involve detection of some form of energy. So the reason you can see is because of light, okay? The reason you can hear is because of sound, okay? And the reason you can smell and taste is because of chemicals, and touch is really some form of pressure, okay? Those are your 5 senses, and they involve energy, okay? They involve detection of some form of energy, and that is how you know what's happening around you, because of your senses. And there's an image there of an elephant supposedly fleeing a tsunami. I forgot what year it was, but in Indonesia some years ago, there was this big tsunami, killed like 70,000 people. And it was reported in some places that people saw elephants moving away from the coast before the tsunami hit. So how is it that they seem to have some advanced warning, that they seem to sense something we can't? Some people say maybe they have some kind of sixth sense, right? You heard of some people talk about a sixth sense. Or maybe they're just more sensitive to vibrations. You know, maybe they can feel the rumbling of the ground when the big waves are kind of, you know, off in the distance, and they can detect the sound waves there, maybe through their ears, or maybe the vibrations through the sense of touch, right, on their feet. So sensitivity of these senses varies. Okay, and yes, there are plenty of animals who have much better senses than we do. Animals that can see better, hear better. Okay, yeah, I mean, there's other senses as well, but yeah, more sensitive to touch and things like that. You know, there are these things out there, but what really sets us apart is our brain, and that will be perception, how our brain makes sense of these information from the senses. But yeah, just wanted to point that out. Let's keep going. Sensation, in the definition, we saw the term sensory receptor. Okay, your senses involve basically sensory receptors. Now, a sensory receptor is just a fancy way of saying that there's a neuron. Okay, a neuron that's involved with the senses. So you have neurons, basically. You have these sensory receptors that respond to a specific kind of energy. So inside your eyes, right, toward the back of your eye, you actually have sensory receptors or neurons that respond to light. Inside your ear, you have some that respond to vibrations. In your nose and tongue, there's stuff that responds to chemical molecules. In your skin, there are neurons there, sensory receptors, that will respond to your sense of touch, which involves some kind of pressure. Okay, so these sensory receptors, these specialized neurons that are involved with the senses, okay, are responsible for what's called transduction. Transduction is merely converting the physical energy, for example, light, right, to the electrical signal. The brain only understands electrical signals. It does not understand the physical energy, light, right? So there's neurons that detect light and then send an electrical signal to the brain, and that's what the brain understands, okay? When the brain makes sense of that electrical signal, that will no longer be sensation. Then it will be perception, making sense of what you're actually experiencing. Okay, so that's a sensory receptor. It's just a fancy neuron. Okay, that detects a certain kind of energy. Okay, and the energy is, and then that's converted to an electrical signal that is sent to the brain. okay? And the sensory receptors are different, okay, depending on, what sense you're talking about, where they are, basically in the eye, the ear, the nose, whatever it is. We mentioned sensory neurons before, as if they're all the same. They're not all the same, okay? But we were keeping it simple at the beginning. Let's talk a little bit more about the physics of sensation. So there is a certain way that these sensory receptors work. They need a certain amount of energy in order to basically send a signal. Certain amount of light energy is needed, certain amount of sound energy for them to fire, to send signals. So there's something known as the absolute threshold to help us understand that. The absolute threshold, it says there, is the weakest level of a stimulus or the weakest amount of energy that's needed to produce a sensation at least 50% of the time. So it's the weakest amount of energy that is needed so you can, let's say, hear something at least half the time. Okay, so if I make a sound, okay, and I would usually do this with the class. I'd ask if you heard that, and if at least half of you heard that, that means that that contained enough energy for it to be detected at least 50% of the time. Okay, but if I make a fainter sound, okay, you probably did not hear that, did not produce enough energy, okay, for you to actually detect that at least half the time. Okay, another example would be the minimum brightness needed to detect a light. The reason we can see anything is because of light. Light reflects off of objects, off of things, and that light hits our eyes, and then the eyes have sensory receptors or neurons that then send electrical signals to the brain. Well, there's a minimum that is needed for you to be able to see at all, for you to be able to detect any kind of light. And if it's below that minimum, then your brain doesn't get the signal. There's a minimum that's needed to at least detect it at least half the time. That is called the absolute threshold, okay? And here's something interesting. Anything below this minimum that's needed to detect something, anything below this, right, is what we call subliminal perception or subliminal awareness, okay? And a lot of you have probably heard of subliminal perception. We're talking about sensation here, but when something's subliminal, that means that you are basically detecting it below conscious awareness. So in other words, your brain is somehow still gets the signal. It's still aware of it, though you may not be consciously aware of that. There's an experiment that was done to help explain this. Experiment done by Karaman, Stroeve, and Claus in 2006. They showed that when Lipton ice is flashed on the screen for 1/50th of a second, okay, so that's very brief, okay? So 1/50th of a second, participants were shown this word like Lipton ice for about 1/50th of a second. Participants did not detect the words. When they were asked, did you see anything on the screen? They said, no, I saw nothing. But then later on, they showed a preference for Lipton ice, which is a type of drink, I believe, type of, iced tea or something like that. They showed a preference for it, meaning that their behavior was affected. Somehow the brain got the message, but they were not aware of it. So when this happens, this is called subliminal perception. Now, whether there is such a thing is debatable. There's some studies that suggest that it works. Some studies suggest that it doesn't work, okay? But it's just something interesting related to the absolute threshold. You need a certain amount of energy to detect something at least 50% of the time. Consciously detect something, okay? If you don't consciously detect it, then you don't know that anything happened. That usually means no signal was sent. Let's keep going. More about the physics of sensation. We also have to know about something called the difference threshold. Just noticeable difference is the same thing, abbreviated JND. So we also need to know the smallest amount of change that can be detected at least 50% of the time. The smallest change in energy, right? So let's talk about taste here. Let's say you taste something and it's in a cup, right? It's some cola drink. And then you taste another cola drink that's in a different cup. Can you detect the difference between those two types of cola drink? Say one is Pepsi and one is Coke. Can you detect the difference? If you can detect that change, that means that there is enough of a change there in some kind of energy for you to at least notice it, okay? For you to detect that there's a difference, okay? I've applied that to taste. okay? But it also applies to vision. You know, can you detect the difference, let's say, between red and green? If not, you know, you might be colorblind in that form. You don't have red and green color blindness. Can you detect the difference between, let's say, one sound and another, right? If you cannot, you might have some problems with your hearing. But we need to know about this thing called the just noticeable difference, or the difference threshold, the smallest amount of energy that you can detect. And when it comes to detecting the smallest amount of energy, it actually varies. Weber's law says that the just noticeable difference is based on a proportion of stimulus change, not a fixed amount of change. What that means is that the amount of difference that you can detect is related to how much difference already exists. So here's an example here that has to do with weight, right? Let's say you can lift 100 pounds, right? Turns out, according to research, you need to add 2 extra pounds for you to know that those 100 pounds have gotten heavier, okay? But if you have 200 pounds, now you need to add 4 pounds to know that it's gotten heavier in order to detect a difference, in order to detect a change. So the amount of change that is needed goes up proportionally, proportionally to what you already have. So to detect a change in 200 pounds, it's no longer 2 pounds. Now it's 4 pounds, okay? And the more weight you have, the more weight you need to add on for you to know that there's a difference there, that it's heavier. Same thing with brightness, right? If it's very bright outside, you go outside, it's very bright, and you turn on a flashlight, you add some additional energy, you can't detect that difference because there's a huge amount of brightness. And what you're adding is not, you don't really detect that difference. But if you're in a dark room where there's very little light, okay, and you turn on that flashlight, it makes a huge difference. The amount of energy the flashlight provided, right, the difference that it makes in the brightness is detectable, okay, because you have a lot less energy, okay? So the more you have of something, the more you need to add to detect the difference. When it comes to sound, taste, with all of our senses. If you're talking about taste, right, and you're drinking some very sweet, let's say, cola drink, right, how much sugar do you need to add for you to know that it's gotten sweeter? Well, these cola drinks, these sugar drinks have a lot of sugar in them, okay? So for you to add a little bit, it's not really gonna make a difference. You need to add a lot more to know that it's gotten sweeter, okay? So that's the difference threshold, or just noticeable difference. And speaking of the just noticeable difference, can you detect the changes in the screen that I'm showing you between the left side and the right side? Can you detect a difference in that color? Or maybe we call it a difference in brightness. One of those sides is brighter than the other. One is darker. Now, if you can, that means that there is a noticeable change in energy there. Okay, a noticeable change in color and brightness, whatever you want to call it. It has to do with our vision. But if you can't really detect it, okay, it's too subtle, the change is too small for you to detect it, that means it falls below the just noticeable difference. And yes, some people's senses are, you know, more attuned than others, more sensitive than others, and they can detect smaller changes, okay? In taste, in vision, or whatever it is. You know, those people who can really detect the difference, let's say in taste, are called super tasters. And it's good if you're a super taster, if you're gonna be a chef or something like that, or a cook, right? Then you can detect the difference between really good food and food that's not so good. But someone who doesn't have, you know, very sensitive taste buds, that's gonna taste good and that's gonna taste good. For them, anything will taste good, okay? They're not very discriminating, okay, is what that means. In other words, you know, they can't really detect a small difference. There is a difference, you know, between, let's say, this meal that's prepared at this place and this meal is prepared at this other place, even though, let's say, it's the same kind of meal. Okay, it's what distinguishes, you know, really high quality food from food that's just okay. But for some people, anything goes, right? They'll eat anything because they can't really detect those differences. Anything tastes good to them. The more sensitive your taste buds are, right, then the better you are at discriminating these things, and maybe the better you are at then being able to tell what's good and what's not good. Okay. This is related also to something called signal detection theory. Signal detection theory is something that is used basically to study people's tendencies, right, toward what we call a hit, a false alarm, a miss, or correct rejection. To put it simply, signal detection theory is something that is used to determine basically how sensitive people's senses are, or how good people's senses are. How good is your hearing? How good is your vision, your taste, or whatever it is? So when you report a hit, or let's say when you get a hit, that's when you say that something is present, and in fact it was. Like somebody, turned on a light, and they asked you, can you see that? And you say, yes, I can see that. I can see that there's light in this room. I can see that it's gotten brighter, or something like that, right? That's called a hit. Something was produced and you detected it. A false alarm is when you report something was there when it was not. It's like they ask you, did you hear that? Did you hear something? And you said, yes, I heard something. I heard a sound. But in fact, there was no sound. That's called a false alarm. It's like when you think you smell gas. Let's say you think there's a gas leak in your home. And so you call the gas company, they send somebody over and they check out your house and they say, you don't have any gas leaks. That's a false alarm. You smelled something, you thought something was present, and it was not. Okay. A miss is when you report something was not there when in fact it was. So a miss would be like basically when you don't smell a gas leak. You don't smell anything. You don't think there's anything wrong. But it turns out that there is a gas leak. And then you try to do some cooking and then there's an explosion and your house catches on fire. Okay, that's a miss. It's when something is present, but you fail to detect it. A correct rejection is when you report something was not there when in fact it was not there. So it's like when you don't smell any gas, right? And you say, oh, there's no gas leak. I don't smell anything, right? And it turns out that there is no gas leak. Let's say the gas company comes over, you double check just in case, right? And they say, yeah, you're right, there's no gas leak. That's called the correct rejection. It's when you say something is not there and it really is true that that thing is not there. Okay. On your homework and on your next exam, you're going to have questions that have to do with this. And the questions might have something to do with, with you. Let's say you're in the shower and you think you hear your phone ringing. Was it ringing or was it not ringing? Or maybe that example with the gas leak. Some example like that. Okay. And you have to know the difference between these. It's a way to test your senses, basically. You can test differences, like they can give you a taste test, right? A cup of one type of cola drink and another type of cola drink. Let's say one is Coke and one is Pepsi. And they used to take a sip of one, take a sip of the other, and they ask you, do these taste the same to you or are they different? And you say, these are different. Okay. And then it turns out, yes, they are different. That's called a hit. You reported something, and in fact, that difference was there, right? And you have to be able to think of those examples, right? With a hit, false alarm, miss, and correct rejection. A false alarm would be basically when, you take the taste test, you take a sip of one cup, then the other, and you say, yeah, these are different. They're not the same. And they tell you, wrong. Guess what? They're both Pepsi. They're both the same. That's a false alarm. You thought they were different. You thought there was a difference there, and in fact, there is not. And you could think of the examples maybe with a miss and with a correct rejection. If you understand how to do this right, then you know what you have to do, right? You'll be able to get those questions right. Here's another way of thinking about it. Not another way of thinking about it, but here's another way of looking at it visually, right? There is, at the top there, you have the stimulus, whether it's present or absent. And then on the side here, you have the report. So when the stimulus is present and you say it's present, that's called a hit. That is correct, right? And when you say something is absent, when the stimulus is absent, right? And you say it's present, see that's in pink. I was going for red there, it didn't quite turn out red. That's an error, that's called a false alarm. That's why it's in red there. When you say something is present, but it's absent, that's another kind of error, that's called a miss. And when something is absent and you say it's absent, that is, you're being accurate there again, that's called a correct rejection. So you can be right in different ways and you can be wrong in different ways. And these things are used to test your senses. Okay, here's another example for you guys. Eyewitness testimony. Eyewitness testimony is, signal detection theory could be applied to eyewitness testimony again. So let's say that you're this, what do they call this comic book guy over here on the left, and those are the suspects for a crime. Somebody committed a crime, okay? And you could be asked, you know, they bring you into the police station and they have the line up there and they ask you, do you see the perpetrator who committed the crime? Okay, do you see the perpetrator in the lineup? And you say, yes, I see the perpetrator in the lineup. And it turns out that it is one of those aliens, then that would be a hit. Okay, if you say, yes, I see the perpetrator in the lineup, and it's none of those aliens, right? Then that's a false alarm. Okay, if you say that, no, I don't see the perpetrator in the lineup, but it is one of those guys, that's a miss. And if you say, no, I don't see the perpetrator in the lineup, the person who committed the crime or the alien who committed the crime, and it turns out that it is none of those aliens, that's called a correct rejection. You have to know these things. It's also applied to white witness testimony, right? We'll talk about memory when we get to that chapter in a couple of weeks. Okay, so that's signal detection theory. Now let's talk specifically about vision. And yes, there's a little bit more biology here, but it's not that bad. And a lot of you have heard about this stuff already. A lot of this is common knowledge nowadays. So let's talk a little bit about vision and how it works a little bit. I'm not going to tell you all the features of the eye, just some, okay? So the eye has this tough, transparent protective layer that's called the cornea, okay? You can see the image there, it's pointing at the cornea, that tough outer layer. It protects the eye, right? It's transparent, it lets light through, okay? Light shines through the cornea and goes through the pupil, which I'm not talking about here, but goes through the pupil, which is the opening of your eye, and then hits the lens. The lens is basically this thing that looks like a disc. It's transparent, okay? Looks like a disc, and the lens changes its shape to focus on objects so that you can see things, you know, that are closer or far away. The lens can change its shape to help you focus. So it's a transparent disc, okay? Kind of like the lens in a camera, okay? That's also a transparent disc, but this lens is more special. The one in your eye can actually change its shape, okay? It can get stretched to make it thinner, right? Or it can be squeezed on to make it rounder, and that helps you see something that's closer or far away, okay? It helps you focus, okay? And then, so the light passes through the cornea, then through the pupil, then through the lens, and it hits the back of the eye. That area on the back, it's called the retina. So it's like the layer of skin in the back inside the eye called the retina, okay? The retina is where the neurons are that respond to light. It contains the visual sensory receptors. Remember that sensory receptors is just a fancy word for a neuron, okay, that responds to one of your senses. So the ones that respond to light are called visual sensory receptors, okay? So the neurons are in the back, right there, of the eye, on that layer of skin called the retina. Let's keep going. So you want to shine the light. The light shines eventually. The light hits back there. It goes through the cornea, okay, through the lens. The lens tries to focus its back there on the retina, where it stimulates those sensory receptors, those neurons, and those neurons are called rods and cones. And a lot of you have probably heard of rods and cones, okay? So the rods and cones are these things that allow us basically to to see, okay? So rods and cones the rods are the ones that allow you to see at night. They're responsible for night vision, okay? And the reason they allow you to see well at night is because they function well in dim light. So those neurons do not require a lot of energy for them to send signals to the brain, okay? And at night, there's not a lot of brightness, not a lot of energy. That's when they work well. And they also, the rods allow you to see in black and white, grays, not a lot of detail, okay? Our night vision isn't very good. So the rods, if you see the image, they actually look like rods. The cones are the ones that allow you to see during the day, and they look like cones if you look at the image. The cones require more energy in order to send signals to the brain. They require more energy to function. So they work well during the daytime when there is more light, okay? And the cones allow us to see color and things in more detail. Our day vision is much better than our night vision. These rods and cones, it says there at the top, are also responsible for what we call light adaptation and dark adaptation. Light adaptation. So let's say you're in a movie theater, okay? You've been watching this movie, and then you know, and it's kind of dark in there, okay? There is some light, but it's kind of dark, okay? And you've adjusted to that light, okay? To that darkness, so to speak. And then you come out of the movie theater, let's say when the movie's over, okay? They turned off the movie, and then they turn on those little lights at the bottom. It's still dark in there, it's dark in there. And then you come out of that movie theater, and let's say right out of the movie theater, right, you make a quick left or right, and you open those double doors and go right into the parking lot. And it's daytime. You were watching this movie in the afternoon. What'll happen as you walk out there in the daytime is you'll be temporarily blinded for a second or two. You will not be able to see anything. It will be just too bright. And then eventually what happens is that your rods will turn off and your cones will turn on. And then you'll be able to see in that very bright environment outside, right? That's called light adaptation. It's also dark adaptation. Let's say you walk into from the outside into a very dark room. Or let's say, this what happens every day. Let's say you're going to bed, right? And you turn off the light to your room. At first, when you turn off the light, you can't see anything. It's too dark. And then you're trying to find your way to your bed, right? Eventually you make it to your bed. Then you're lying there. And a few minutes later, you realize that, hey, I can see now. That's called dark adaptation. What's happened is that, you know, when you turned off the light, your cones didn't work anymore because first you had the light on and the cones were working. So you were using your cones that require a lot of energy. And then you turned off the light and now your cones don't work. You can't see. You need to switch to your rods, and it takes a few seconds. Actually, it takes a little bit longer. It might take a few minutes for your rods to turn on sufficiently enough, or enough of them to start working, that now all of a sudden you can see at night, right? You can see in the dark. But there has to be some light, a little bit of energy, right? You will not be able to see anything if it's completely dark, okay? Your rods won't work either. They require a little bit of energy. So the rods and cones are responsible for night vision, day vision, light adaptation, dark adaptation. Okay, a lot of you already know that. Okay, now I told you guys that the light shines through the cornea, right? Goes through the pupil, through the lens, right? And then hits the back of the eye, the layer of skin back there called the retina that contains the rods and cones. The lens actually tries to focus the light, not at any part in the retina, but in the center right there where you have the fovea. Okay. The fovea is the center of the retina. The eye tries to focus the light back there because that provides the clearest, sharpest vision. So daytime, right? You're trying to see something. For you to see really well during the day, right? Light enters the eyes, goes through the cornea, through the pupil, through the lens, shines on the retina in the back, and it shines on. If it shines on the center of the retina, if you can focus the light there, that's how you'll see best. Okay, so your lens is trying to focus the light at that spot. Some of you, your lens isn't very good. You need glasses to focus the light there. Okay, you need corrective lenses because your lens doesn't work very well. Why is it that you see best if the light is focused at the fovea? And by the way, the center of the fovea is also called the macula. Okay, so it's like the center of the center. Your book might mention the macula. I'm just keeping it at the fovea. Okay, that is where most of your cones are, at the actual fovea. So for you to see very well, you need to focus the light at the fovea, where you'll stimulate the greatest number of cones that allow you to see in color, in detail, right? You can see well during the day. At night, they don't work very well, okay? So at night, actually, you can see best by actually focusing the light a little bit toward the edges where the rods are, okay? But night vision is different, okay? But the most cones are in the fovea. As you go away from the fovea, away from the center, and toward the edges, you will have more rods, less cones and more rods. So if you want to see really well, especially during the day, right, you want to focus the light at the fovea. Okay, that's where most of the cones are. Turns out you also have a blind spot. There's a point in the retina where there's no rods, no cones, none of these visual sensory receptors. And if you shine the light at that place, you'll see a blind spot. You'll see a point that will just look like air. It looks like things are invisible, right? That is where the optic nerve connects. There's no rods and cones there. You can see the optic nerve over here on the right, that thing that looks like a wire, and it continues, right? if you shine the light there, there will be a blind spot there. It will look like you're looking through air. There is a way to find your blind spot, but I think your book doesn't have that demonstration. So I don't have it here either. But each eye has a different blind spot, by the way. So they cancel each other out. In order to find your blind spot, you need to close one eye. And then you have to do some little demonstration. Maybe you can find something online. I keep thinking about it, but I haven't put something in here to help you do that. And we wouldn't do it remotely anyway. It doesn't work remotely. We could do it in person, right, in class. But we're not doing things that way right now. The optic nerve carries the visual information, the electrical signal, to the primary visual cortex, which we already talked about in the previous chapter. And that is the part of your brain that understands vision. Okay. Let's talk about something related to vision that's more interesting. The theories of color vision. How is it that we see color? Okay. The simplest theory of how we see color is the Young-Helmholtz theory, also called the trichromatic theory. Young-Helmholtz, named after, I think that was one person, or maybe it was two people, and that's why it's Young-Helmholtz. Sometimes people have a hyphen in their last name, but either way, it's named after someone or some people, right? Or trichromatic theory. Tri means three, chrome has to do with color. It's a theory that basically says that there's three different colors that you can detect, and by mixing these three different colors, you get all the other colors, okay? A little bit more complicated than that. It says we have three different, well, not really that more complicated, but the word is a little more complicated. Photopigments, okay? There's three different colors that we can detect. Mixing all these colors gives you all the other ones. So so you can see there, they're red, green, and blue, okay? And I believe that these are called your primary colors, or maybe the primary color. You know what, I remember seeing the song on Sesame Street, and I don't remember if it's exactly these colors they're talking about. But this theory says there's three different colors, right? Blue, red, and green. And by mixing these, you get all the other colors, okay? So if you mix red and blue, you get violet. If you mix green and blue, you get, I guess, some kind of light blue. If you mix red and green, you get yellow. And if you mix them all together, you get white. Now, when you mix paint, it doesn't really work this way, okay? Because when you mix paint, it doesn't always work. Well, if you mix all that paint, right, like red, green, and blue, you actually get something that looks like brown. This works with light. Remember, your visual system works with light, okay? And when you do have different colored lights, when you mix them all together, you get the white light. White light actually has all the colors. When you spread them out, you can see all the different colors. And that's what happens when you see a rainbow. The light is being split into all these different colors. The white light is being split into all the different colors. So that's what the Young-Helmholtz theory says. It says we can detect three different colors, and by mixing them, you know, we get the other colors. And of course, this happens in the brain, okay? Now, there are people who are colorblind who only can see two of these three photopigments. They can only detect two of the three colors. Like what's being portrayed here is red-green color blindness. So there's people who have red-green color blindness, basically, and you know, they do not see the world the way most of us see the world, okay? So a normal person can see things like the top two images there, right? They can see, you know, the guy's shirt there is red. They can see that painting of the sunflowers by Van Gogh, right? They can see the painting the way it really is. But someone with red-green color blindness doesn't see things that way. You can see in the image on the bottom, right? That person there on the lower left, right? They can't see the red. They can't detect the red, okay? And in the sunflowers, they can't detect the red either. Instead, things look green. So this person would have red-green color blindness. So they are missing two of the photopigments. Here's the thing. So you have, the theory really says you have three different kinds of neurons, okay, that detect three different colors. And if you're missing one of those neurons, then you have color blindness. If you're missing, you know, one of those, you might have red-green color blindness. By the way, if you're missing two out of the three neurons, if you could only detect two out of the three colors, let's say, or if you're missing two of the neurons for the three colors, what happens, it's even worse. What happens there is you can't see color at all. You can only see like black and white, okay? So that's the theory, right? There's another more complicated theory, which I think is a little bit more accurate, and that's opponent process theory. Opponent process theory says something a little bit more complicated. Opponent process theory says that the human visual system has 3 different types of receptors. So like 3 different kinds of neurons, okay? But now, rather than those neurons detecting just one color, the neurons actually detect one color and its opposing color, okay? So there's a neuron, for instance, that allows you to see red and green, or you could say red or green. There's another that allows you to see blue or yellow. Another one that allows you to see black or white. And depending on which ones are on and off, you'll get the different colors. The simplest one to explain here is black and white. Okay, if basically all the neurons, all the 3 receptors for these, the red, green, the blue, yellow, the black, white, if they're all 3 of them are off, then basically that means there's no light. You see black, okay? Or all the light is being absorbed, okay? Now, if they're all on, you get all, all the colors, right? That's white. And you can see the pattern there for when you get red, when you get green, when you get blue, when you get yellow. It's a little bit complicated, and I'm not gonna explain it for the sake of time, but I will show you something, a demonstration that you can do that we usually do in class. But this basically, to explain it, just make it a little bit simple. This theory says that you have 3 different types of receptors. You see red or green, blue or yellow, black or white, okay? So each color has its opposing color, okay? And you see one or the other, okay? And that may not make much sense right now, but take a look at this. If you're paying attention, take a look at this, right? What I want you to do there, and I would do this in class, and it works really well in class. I want you to stare at that white dot for about 30 seconds. Just stare at it. Keep staring at the dot. Let's say it's been 30 seconds, okay? Now what do you see here? It works much better in person, but as you can see now, this is just a blank screen over here, a blank slide. There's nothing there. But when I showed you this and you stared at that white dot for about 30 seconds, a minute or something like that, and you really just focus on that, when I show you the next screen, what you see are the opposing colors. So where there is black, you saw white, so the stars appear white. Where there's yellow, you see blue, right? Where there's green, you saw red, and where there's black, you saw white. So if you did it right at this blank screen over here, you would see an after image. You would see the American flag with its true colors. Those are the opposing colors of this image right here. And that's kind of how opponent process theory works. Now, here's another image for you guys. Some of you may remember, maybe are old enough to remember, there was a time when there was this dress that caused quite a stir, right? There were reports that there's this dress, right, that appears to be different colors to different people. That some people see the dress as white and gold, and some people see it as blue and black. So if you're looking at this right now, to some of you, it appears blue and black. That's the way I see it right now, by the way. And to some of you, it might appear white and gold. Why is that? It's a little bit of a mystery as to why it happens. But according to a vision expert that I talked to, it has something to do with the background. That the background basically has basically affects your perception of color a lot. So a color can seem very different, a lot brighter or darker, if it is next to a certain color. But there is something here that's going on that's kind of like opponent process theory. Because for some of you, right, this is blue and black, right? And for the rest of you who see white and gold, where the rest of us see blue, you see white. Now that's not quite the opposing color. The opposing color should be, yellow or something like gold, right? It's white instead is what you see. It's not quite the same color, but it's close. For the rest of you, and for those of us who see black, the lacy part there, where it's black, right? Some of you there see gold. Now, the opposing color of black is white. Gold is not quite the same thing, but it is a lighter color. So there's something like opponent process theory that's happening here that's going on. But it also has to do with the background. There's several things going on here. Color has a lot to do with basically what else is happening around you. Something can appear basically dark gray or light gray. It can appear white or a different color, depending on what else is happening. And it's kind of like opponent process theory. All right, that's it for vision. We talked a lot about vision. Now let's talk about hearing. We're going to go through the other senses much more quickly, okay, to make sure we finish on time. But hearing, right? Remember, we start talking about things at the biological level, okay, and physics a little bit, right? The reason you can hear is because of a type of energy, right, that's detected by sensory receptors, right, and electrical signals are sent to the brain. Well, the type of energy that's detected, they're called sound waves. They're waves, basically. Waves can travel through the air, they can travel through water, they can travel through solids, and depending on how solid something is, they might not travel very well, okay? If the object is somewhat solid, like a wall, let's say, and you talk loud enough, somebody might be able to hear you on the other side, but a lot of waves get blocked, so you don't hear them very well. Now, if you have a very solid object and you yell, the waves will bounce back at you and you get an echo. But basically, it's sound waves. That's the reason why you hear. Sound waves travel through the air, through water, or through solids. They hit your ear, and inside your ear, you have these sensory receptors that are called hair cells, right? Those are the sound receptors in the ear. They detect those waves, and then they send signals to the auditory cortex, the part of the brain that understands hearing. Now, I skipped over the characteristics of the wave. Waves have different characteristics. There's the frequency of the wave. That's the number of cycles per second. The frequency of the wave corresponds to pitch, and that's measured in hertz. When you have a high-pitched sound, you know, that would be like basically birds chirping, you know, or little kids have high-pitched voices, right? Low-pitched sounds would be more like the roar of a lion or someone with a very deep, manly voice, right? Frequency is what gives you that, right? Frequency has to do with the number of cycles in a second, right? In a wave, right? If you have a wave that looks very compressed, very tight, okay, a lot of up and down, you know, in a short space, right? That means it has high frequency, high pitch, or high energy. The waves that are a bit longer, that are a bit more spread out, right? Those have lower frequency that corresponds to low pitch. The amplitude or the height of the wave corresponds to the loudness, okay? And then timbre, something, right, that has to do with the complexity of the wave, that corresponds to the qualities of the sound. That's a little bit harder to explain. If you're a musician, you probably understand timbre a bit more, but you know, it's like when you combine several notes, let's say on the piano, and they create, rather than having a simple wave, you get a complex wave. That's kind of like timbre, okay? But I'm not going to try to explain it. I'll ask you more about frequency and amplitude. Here's an image for you guys so you can see the wavelength, right, is basically the wave from one peak to another, okay? If the wave is longer, that usually means that it's basically lower frequency. If the wavelength is shorter, that's like a tighter wave, that's higher frequency. The height of the wave is the amplitude that corresponds to loudness. Okay, same thing I just said, it's just an image here of a wave. Okay, if this was a higher frequency wave, meaning it corresponds to higher pitch, we'd have to take the wave and squish it on the ends and make those peaks, right, like those little mountains, come closer together. Okay, and that would be higher frequency, a higher energy wave. Let's keep going. Here's an image for you about how the ear kind of works. I'm not going to go into all the complications, but I'll tell you more about the things that are actually in there. So sound waves travel through the air, okay, and then they hit that flap of skin that you call your ear. Turns out that's only a part of your ear. That's only what we call the outer ear. That's only part of the outer ear. So the flap of skin, it's called the pinna, and then the sound waves going through that hole in the middle of the ear, that's called the auditory canal. The auditory canal and the pinna make up what we call the outer ear. So the sound waves hit the ear, right? They hit the, you know, that fleshy structure, go through the, you know, the auditory canal, right? That's the outer ear. And then the waves will eventually reach what we call the middle ear, which are three little bones, okay? The malleus, incus, and stapes, right? They hit these, the sound waves will basically, first they'll reach the, you know, what's called the eardrum, okay? And that's connected to the, basically these three little bones, okay? That is what we call the middle ear. So it's like the sound waves are now kind of pushing through those, solids, okay? Those three little bones. And then those little bones will then push on the little thing that looks like a snail. It's called the cochlea, okay? The cochlea is what we call, it's part of the inner ear, okay? The inner ear, the cochlea, and the basilar membrane, which is, you know, some tissue that's inside, okay? So that thing that looks like a snail is the cochlea. It's filled with fluid, right? So when the little bones push on it, it creates waves in there. And then the waves cause other things to move. The hair cells are in there. It causes them to bend. And when they bend, they send signals to the brain. Okay, and then the brain makes sense of it. A lot of complications here. I just need you to know a little bit about how sound works, and that information is right here. You need to know about hair cells and frequency and amplitude, all what that corresponds to. Like I said, we're going to go faster through these other senses. Vision is the most interesting. We're primarily visual. Okay, that's the most important. Olfaction, your sense of smell. Okay, that's scientifically it's called your sense of olfaction. Now, the way the smell works is that there's these molecules that are floating around in the air. Those are called odorants. Okay, they're floating around the air, and you take a whiff. Some of them go inside your nostrils, and inside your nostrils, inside your nose, there are these things called olfactory receptors. Neurons, basically, that detect those molecules, or they attach themselves to the olfactory receptors, right? And then they break down, molecules break down, release energy, and then an electrical signal sent to the brain. These olfactory receptors are on a piece of tissue called the olfactory epithelium. So inside your nasal cavity, inside, you know, the nasal cavity, which actually is a big space inside your face there, there's this piece of tissue from which, you know, these olfactory receptors hang down. And you see the image right there toward the bottom there in the little square, right? You have the olfactory bulb, which sends the electrical signal to the brain. And then hanging from there, attached to that, are the olfactory receptors, right, that hang down, you know, through the, basically, the olfactory epithelium, which is just a piece of tissue. They hang down into, basically, the nasal cavity, into the space that's inside your nose and further in your face. They hang down in there, and when you smell something, the things go inside your nostrils, and they attach themselves to those things. They break down, release energy, and an electrical signal is sent to the brain. Some of that information, the electrical signals are sent to the limbic system, which has to do with emotion, memory, that kind of stuff, right? Because smells, yes, can trigger emotions. They can also trigger memories. And some of the information also goes to the primary olfactory cortex, which we didn't talk about before, but it distinguishes odors, right? It allows you to understand what odor that might be, what thing that might be, right? And sends other information to other parts of the brain, because the brain also needs to respond to these things. If it's something disgusting, for instance, or if it's a, you know, a certain pleasant smell, you want to approach it. So that's how basically olfaction works. Okay, I would always know what the neurons are called for the primary sense of, for the primary sense, right? For that sense, right? Know you know, know what the scientific term is for that sense, olfaction. Know a little bit about it, okay, and you'll be okay. You can see here an image. You can see, right, the nasal cavity there. This big gap in your face, right? Those little things that look like hairs are called the olfactory receptors that hang down from that piece of tissue. And also the olfactory receptors also are attached to the olfactory bulb, which sends the signals to the brain. Okay, so when you take a whiff, stuff goes in there, attaches itself to those little things, the olfactory receptors that have these little things that look like hairs, right? That's called cilia. And, you know, that attaches itself there. The molecules break down, release energy, and then a signal is sent to the brain. Okay, that's the way smell works. I know I'm going a little bit quickly, but I want to make sure we get through this. Now, the sense of taste is a little bit more interesting, but we're going to keep it simple. Believe it or not, there are only like 6 different kinds of taste that we can detect. We can detect things that are sweet, You know, we love things that are sugary, or fruits that are fruit that is sweet, right? But we can also detect things that are sour. Oh, by the way, things that are sweet usually give us quick energy, right? Simple carbohydrates, it's important that we detect that. Things that are sour, usually, basically that has to do with what we call citric acid, which is also vitamin C, right? Which is found in lemons and in, you know, in oranges and things like that. It's important for us to detect sour things, okay? Because of vitamin C. Salty things, we love things that are salty, okay? When you add salt to food, it makes it taste better, okay? That's one of the types of taste that we can detect, okay? Without salt, by the way, food doesn't taste very good, okay? It's just, it doesn't have much flavor. Flavor, by the way, is more complicated. I'll tell you about it in a little bit. Taste is different. There's only about 6 different kinds of taste. We can also detect things that are bitter. Things that are bitter, that usually means that something is poisonous. Like if you try to chew some leaves, they'll probably be bitter. They're poisonous to you. You're not supposed to be eating those, okay? Drink something that, which I don't recommend, by the way, something that's poisonous, whether it's gasoline or some other thing that's poisonous, it'll be very bitter to you. And that usually means it's poisonous. That means it's something you need to stay away from usually. There are some things that are a little bit bitter that some of us like broccoli is a little bit bitter, by the way. But some people like broccoli more than others. Turns out that those who actually like broccoli more are the ones who cannot detect the bitterness that much. Those of us who hate it, who don't like it very much, we can detect the bitterness a little bit more. So we don't like it that much. When something is bitter, usually indicates that it's probably poisonous, or it has a little bit of something that's poisonous. Broccoli doesn't have enough of it, so it's not really bad for you. It's actually good for you, but it's a little bit bitter. And then there's the taste of umami, which is monosodium glutamate. That's what MSG stands for. Umami was discovered by the Japanese. That's why it's called umami. And it usually has to do with flavor, not with flavor, with taste that is kind of meaty, savory, kind of things that have protein in them, milk, cheese, meat, eggs, that kind of stuff. That's umami. And then there's the fatty taste, right? We also like things that are fatty. When things have a lot of fat, they usually taste better, okay? Those are the 6 kinds of taste that we can detect. In the past, we only knew about 4. When I started teaching, there was only 4. And then they added another one, then there was 5. Now there's 6. There might be others that will be discovered in the future. But as far as we know right now, there's only 6. So we can detect those 6 different kinds of taste. And by the way, your sense of taste is called gustation. You need to know that. And there's papillae, there's bumps on your tongue that contain your taste buds. And then the taste buds contain your taste receptors, the neurons that respond to taste. Right? When you eat things, they break down, release energy, right? Taste receptors detect those chemical molecules. They send information to the brain via send information to the brain to the gustatory cortex, which distinguishes the different kinds of taste. Okay, and that's all you need to know about taste. Now let's talk about your sense of touch. Touch is actually very complicated, but we're going to keep it simple because this is a 101 class. And today in talking about sensation, we're talking more about the physical things about sensation. When we talk about the mental stuff, how you make sense of it, that'll be perception. That's on Wednesday. So to keep it simple, there's 4 components of touch. There's 4 different kinds of touch. There's pressure, warmth, cold, and pain, okay? Thermoception refers to hot and cold, okay? Nociception refers to detecting pain, basically. And then touch is just pressure. Now there's all these different things that are in the skin. The reason you can, the reason you detect anything through touch is because of your skin, okay? You also have skin inside your body, but it's not called skin, but you have organs and things inside your body. And when something's wrong in there, or you can actually feel certain things in there, like you have a broken rib like I did, or something wrong, you have some injury, some bruise, some internal bleeding, something like that, you can usually, there's some, you can also detect those kind of things. But usually stuff inside the body, you're used to it, but you can also feel things in there. So, but we're going to talk primarily about the skin, okay? The skin on the outer part of your body. It has all this stuff in it that allows you to detect different aspects of taste, of touch. It has these things called Merkel's discs that allow you to detect light pressure. It has Ruffini corpuscles, allow you to detect, when something stretches, like if your skin stretches. Mesner's corpuscles that allow you to detect pressure, right, and lower frequency vibrations. There are others, just I don't put them all in there because I don't want to give you too much information. There's other things that allow you to detect, you know, basically things that are itchy, like when something makes you itchy, it makes you want to scratch yourself, right? There are things that have to do with that. There are cold receptors. There are neurons that respond to cold, okay? That when the temperature drops below a certain level, these neurons send signals to the brain, and then the brain knows that it's cold. Or other neurons that fire when temperatures are above a certain degree, when heat is above a certain temperature, right? And then the warm receptors fire, and then the brain knows that it's hot. Nociceptors, they detect information about pain and injury. And then the information is sent to the somatosensory cortex. Interesting thing about warm receptors, about detecting heat, is that if the heat goes beyond a certain point, I don't remember exactly what temperature it is, but it might be around 113, 115 or something like that. When the temperature gets that high, Fahrenheit, warm receptors, what happens when it's that hot is nociceptors will also begin firing and sending signals to the brain. And what that means is that it's so hot that it's painfully hot, that you are actually burning, okay? And it takes a while actually for you to start to burn, depending on the temperature. But if you stay out there long enough and it's hot, you'll begin to burn. And the higher the temperature, the less time it takes for you to burn, okay? And when you start to burn, and you start to get, it becomes painfully hot, that's when the nociceptors are kicking in. But they also, you know, have to do with injury and other things, right? Other types of pain. Those are the 4 components of touch. I need you to know about these. I put the stuff in color that I want you to know about, okay? The thermoception, the nociception, cold receptors, warm receptors, know about that stuff. And here's an image for you guys, okay? So you can see some things on the skin there, okay? Skin surface, that you can see what Merkel's discs look like, the Ruffini corpuscles, the penicillium corpuscle, which I didn't mention, right? The Mesner's corpuscles, all these different types of things inside your skin allow you to detect different things, whether it's being itchy, light pressure, pain, vibration, things like that. There's different things that detect, different things in the skin that allow you to detect different things, okay? And the next part is about perception, which we will save for later. Okay, we'll save that for Wednesday. So I'm going to stop recording now.